Perfect. Okay, while we are continuing to let people in from the waiting room, I'm just gonna get started with some introductions and some housekeeping. And as people filter in, we'll let people filter in as we're doing the introduction so no one misses the meat of the presentation. So thank you everyone for joining our webinar today. We're going to be talking about innovative drug delivery platforms for MPTs and beyond. We've got 4 really interesting speakers today that we're really excited by. We wanted to let you know that the webinar will be recorded and available on our website. We did have a question about that, so wanted to make sure everyone knew that it is being recorded, and it'll also be available if you're not able to stay for the entire time. So if you could go to the next slide, please. A brief overview of what sort of the structure of the webinar. We're just going to have a brief overview of sort of the IMPT, the initiative, and sort of what MPTs are for anyone who is not familiar. We've got, like I said, 4 featured discussions. We have Dr. Alessandro Grattoni, who's the Frank J. and Jean Raymond Centennial Chair and Professor of Nanomedicine at Houston Methodist. We have Patrick Stayton, who's the Distinguished Term Professor and Director of Molecular Engineering and Sciences Institute at the University of Washington. We've got Dr. Oleh Taratula, who's a professor at Oregon State University College of Pharmacy. And finally, Dr. Kevin McHugh, who's an assistant professor of bioengineering at Rice University. And I'll give a more a high-level summary of the research they're doing and a very top-level summary of their presentations today. Following their presentations, we're going to have a facilitated discussion. We have some questions that we've already received, and then we will also open the floor up for discussion. While we're going through the introductions, if you would feel free to put your name and organization affiliation in the chat so we can get to know you, that would be great. And then finally, we'll just wrap up and adjourn and just give you some resources where you can find more information. So if you go to the next slide, please. So this webinar series is brought to you by the IMPT, which is the secretariat that is working on advancing and making sure that MPTs continue to receive support, funding, and coordination across the field. So our mission is to foster an enabling environment for the successful advancement of MPTs for women around the world. And if you are not familiar with MPTs, they are multi-purpose prevention technologies that aim to provide contraceptives along with either HIV or other STI preventatives. If you go to the next slide, please. So one of the things that we do at the IMPT is we house a MPT product development database, which is available on our website. It's actually in the process of our annual update right now. So if you see anything that's out of date, if you're familiar with anything, please let us know. And when you do hear from our staff asking for updates, please send us that information so we can make sure that this is up to date. And then if you go to the next slide, please. I just wanted to show a very quick overview of one of the resources that we have on our website, which is, you'll see at the top, mpts101.org. And so as part of our database, you can filter by indication and delivery type. And this is particularly important and interesting for our talk today because we're going to be talking about new drug delivery platforms. So as you can see, there's a wide range of different MPT platforms that are under development. We have a lot of intravaginal rings, but we have a couple different other things. We've got orals, injectables, films across the board. And so while we do know that choice is really important for women and providing a lot of different product types is important, we're always looking to see what else is out there, what other kinds of technologies and platforms and delivery types we can be using. So that's why I think this is going to be really interesting. So without further ado, I wanted to introduce today's discussants. If you could go to the next slide. So our first speaker is Alessandro Grattoni. His research focuses on the development and clinical translation of implantable nanofluidic technologies for drug delivery, molecular sieving, and cell transplantation. His lab has developed silicone-based nanochannel membranes for the controlled delivery of therapeutics by exploiting nanoscale physics, achieving a constant or tunable sustained release of molecules for extended periods of time. So today he'll be discussing his research around an ultra-long acting refillable implant as an MPT. He's also been working collaboratively with our second speaker, who's Dr. Patrick Stayton. His research is focused on developing new anti-infective therapeutics, immune therapies, and drug delivery systems. His lab has developed a new polymeric prodrug platform for long-acting injectable or implant-based HIV PrEP and therapy, and as well as contraceptive applications. So our first two speakers are actively working in the MPT space, and then we'll be followed by two more speakers who are not currently working on MPTs, but are doing some really great innovative research that we thought might be informative to this field, and is honestly just really interesting science. Dr. Oleh Taratula will be our third speaker. His research focuses on novel nanomedicines for imaging and treatment of cancer, endometriosis, and other diseases. He is working on developing multifunctional nanoplatforms for fluorescence-based surgery, intraoperative phototherapy, magnetic hypothermia, and gene therapy. So while he's not currently investigating MPTs, he'll be talking specifically about his research in developing targeted therapeutics to the female reproductive system, which is sort of its tie into our interest in MPTs. And then finally, our last speaker is Dr. Kevin McHugh. His primary research interest is in developing a biomaterial microdevices for drug delivery and tissue engineering. His lab combines cutting-edge manufacturing techniques such as multiphoton 3D printing at the nano and micro scale for favorable material properties to facilitate device behavior. By using polymers' well-understood structure-function relationships, Dr. McHugh aims to rationally design constructs that demonstrate predictable behavior and can be customized for a variety of applications. So we're going from MPT-specific research to sort of more research that's a little bit more sort of agnostic around its specific application. We've asked each speaker to talk for about 10 minutes, which is not even close to enough time to cover everything they're doing, but we wanted to allow everyone to give an overview. So with that, we'd like to turn it over to Dr. Grattoni.
Hey, good morning, everyone. I'm delighted to present today a little bit of the work that I've been doing collaboratively with Dr. Patrick Stayton. If you guys could please share my slides. Thank you. So I will present today about our ultra-long-acting refillable nanofluidic implant that we have originally developed for HIV prevention and potentially treatment, and we realize that has potential in the context of multi-prevention technologies. Next slide. Before I jump in straight into the technical presentation, just want to let you know where we are located. We are in the heart of the Texas Medical Center in Houston, Texas, which is a phenomenal environment for biomedical research with all the different research institute and hospital that are surrounding us. So we can find the expertise in any sort of field around us. This is a picture in the bottom right. There is a picture of my laboratory. We have been here at Houston Methodist Hospital for about 15, 16 years. And we're looking forward to actually try to make an impact in the field of HIV prevention. Next slide. So a few years ago, about 20 years now, we started working on nanofluidic system for controlled drug delivery. My laboratory has been focusing entirely on the prevention and treatment of chronic diseases across a broad field of applications ranging from metabolic diseases, infectious diseases, as well as cancer. And we have developed silicone microfabricated membrane with regular arrays of nanochannels, all identical to each other, with a very clear monodispersed distribution around the surface area, and generated with the same technologies that are used for fabricating microcomputer chips. We have been able to demonstrate that by using nanofluidic system, we have the ability to control the transport of molecule from, for example, an implantable reservoir into the body fluid for extended period of time. So our system, in simple terms, resemble an hourglass where the nanochannel themselves operates as the friction in the glass that control the release of the molecule until the molecules are completely eliminated. Next slide. One of the interesting features of our implant that kind of distinguishes from other implantable technologies that have been developed in the literature, been shown in the literature, is the fact that it's transcutaneously refillable. So we do understand, and also from an end user acceptability, that the required procedure of implantation and explantation of drug delivery implant is not great. Obviously, nobody wants to go through a repeated procedure, an intervention that requires a surgery, despite the surgery being minimal. So we realized that there is the opportunity to have these devices working for long-term application, meaning for potentially tens of years, once implanted underneath the skin, and delivering drugs for extended period of time. We are planning drug delivery, continuous uninterrupted drug delivery in the order of 2 to 5 years, and potentially refill them just transcutaneously. If you don't mind clicking on the video, there should be a little demonstration of our procedure for refilling. You can appreciate this is an implant that is inserted underneath the skin in a rhesus macaque, and the procedure that has been performed at the moment is really the refilling of the drug. So the implant is loaded and inserted underneath the skin empty, and then at the time in which we want to initiate the therapy, then we perform this very rapid procedure that takes literally less than 30 seconds to be performed, and in a clinical setting could be performed in a doctor's office, both for the initiation of the treatment as well as for the replenishment of the drug once the drug is being completely exhausted from the device. Next slide. So after transcutaneous refilling, you can appreciate the device is fully loaded. In the inset A, you can see a device that it's being transcutaneously refilled. Part of the secret sauce for our refilling is using particle suspension. So we use drugs in a solid form, suspended in a media, and during the procedural refilling, the particle remains trapped inside of the implant where the media is capable to escape through a filter that is placed at the outlet ports of our device. So this allows us to maximize the amount of drug that is loaded within the implant and therefore minimize the volume of the implant itself. We have shown in our studies that the refilling procedure doesn't affect the pharmacokinetics, and despite the fact that the implant is being inserted under the skin for an extended period of time, doesn't really affect its function. So we are capable, refilling after refilling, to recapitulate and regain the same level of pharmacokinetics that we were observing with the previous iterations. Next slide. So we did a lot of work in the context of HIV prevention. We first started working with tenofovir alafenamide fumarate from Gilead Sciences. Unfortunately, part of the work was halted because of some of the issue, the concern that several groups have reported in the literature regarding the potential inflammation, local inflammation of tenofovir alafenamide in tissue surrounding implantable devices. We move forward into examining the potential of another antiretroviral drug, islatravir from Merck. Islatravir is a different type of molecule, definitely in higher stability at body temperature and in presence of biological fluids, which we evaluated to be over 3 years in that implanted environment. So we felt it was an ideal candidate for HIV prevention and potentially for multi-prevention technologies. So we look into pharmacokinetics analysis in rhesus macaque in 4 animals in particular. And we follow with one single administration of this implant. We follow the pharmacokinetics for 29 months. And as you can appreciate in panel B, we had the ability through this implant, nanofluidic implant, to sustain concentration at constant level for the same amount of time without really an inflection. So this was indicative of multiple things. One of the questions that we typically ask is whether these implantable devices inserted subdermally, once they are enveloped by a fibrotic encapsulation, which is typical of any sort of implantable device, whether their function is in fact affected. And as the data, the pharmacokinetic data that we're showing that demonstrates definitely the fibrotic encapsulation that we have surrounding the device has no appreciable effect on the pharmacokinetics. Therefore, we were very pleased to see this. Once again, this is 29 months of sustained release with no interruption and no refilling. We estimated with the implants that we had developed and that I showed you just in the previous slide, we could have continued delivering islatravir continuously for 13 years uninterrupted, given the fact that this islatravir dose required for HIV prevention is relatively small, approximately 100 microgram per day. So with the same technology, we showed that not only we had the plasma level of islatravir, but of course also the metabolite, islatravir triphosphate, had surpassed the preventative level. And one of the key aspects, also another question that we typically receive, is how quickly do you achieve with your implant the preventative level of your drug? And so in this case, using these 4 monkeys, we were able to see that all 4 monkeys were above the preventative level only 3 days after implantation of the device. And in fact, 3 of them, 3 out of the 4, were above preventative level or already at 24 hours post implantation of the device. Next slide. So we proceeded and we performed a study examining the preventive efficacy against SHIV infection in NHP. We used 2 different animal cohorts. In male, we perform low-dose repeated rectal challenges, and in female, low-dose repeated vaginal challenges of SHIV-SF 162P3. And as you can appreciate from the Kaplan-Meier curve, we were able entirely to prevent SHIV infection in this animal, while the control got all infected within a relatively small number of challenges, a total of 5 challenges for all the control being infected in the male animals, while 2 challenges were sufficient for the vaginal study group. So we were very pleased with the results, and we're hoping to developing this technology further, even in collaboration with Merck. Next slide. One of the key aspects that in fact, many of you may be familiar with is that clinical trials with islatravir in the context of HIV prevention and with the implant that Merck had developed were halted due to the fact that there was a decreased number of lymphocytes that were detected in the clinical trials. But the clinical trial that detected this decrease in lymphocyte was CD8 positive T cells, were specifically related to oral delivery of large doses, so once weekly, once monthly doses of islatravir. We realized that in our models, we didn't have any sort of inflection in the lymphocyte count, and not even on CD4 positive, CD8 positive cells. Neither we had any sort of adverse effect in terms of toxicity, creatine level, AST, ALT level. Next slide. We paid particular attention because of our experience with tenofovir, we paid particular attention at looking at the local tissue surrounding the implant. What we are doing is continuously dosing a certain amount of drug in the surrounding, the proximity of the device. And so that part of the tissue that it's receiving the drug at a higher concentration before it's actually uptaken by the systemic circulation, it really needs to be evaluated to make sure that no inflammation, no chronic inflammation is developed. So again, separating male versus female, because we understand that the response to foreign body response to implantable devices is actually distinct. We were able to see that in both cases where we had islatravir, we had an increase in the thickness, a thickening of the tissue surrounding the implant for the first 15 days after initiation of islatravir treatment. But that actually normalized and relaxed, and after that, the level of the tissue in terms of thickening and the inflammation, potential inflammation, was completely resolved and was maintained for the rest of the study. From an histological score, we definitely saw that the effect of islatravir gave a little bit worse scoring as compared to saline solution devices, but all of these were within still an acceptable range that would consider this device acceptable for long-term, ultra-long-acting application. Next slide. So obviously, in the context of MPT, what we decided to do was focusing on HIV prevention and contraception. We started working with the tonogestrel, and straight out of the bat, we decided to take a tonogestrel the way it is and load it inside of our device without any sort of formulation. As many of you appreciate, the tonogestrel is highly hydrophobic, and we realized soon that the release rate that we were achieving was way too low, 1.5 approximately microgram per day. Obviously, it's not sufficient for the intended use. We were targeting, and we are currently targeting 35 microgram per day release. So we started working on formulation. Unfortunately, I cannot share the details of the formulation themselves, but with the first attempts, we weren't really successful of increasing substantially the drug release to the point that it was suitable for clinical application. But lately, on the graph on the right, we are targeting a complete different formulation approach with different type of excipients, and we were able to see that we are right on target, perhaps even exceeding a little bit the concentration needed. However, obviously, as you can appreciate, this is only first 3 days of release. We typically have sustained zero-order release that continues for months and years. However, still, this needs to be verified. So this is very early data set that have not been published. Next slide. Now, I'm very excited to work on this MPT program with Dr. Patrick Stayton. I met him first at the Bill and Melinda Gates Foundation a few years ago, and I immediately, upon his presentation, I fell in love with his work. Definitely, you will see a lot more from his presentation right after mine. His work on Dragamer, on polymeric formulation of drugs, really clicks very well with our implant technology. We understand that the implant technology offers a protected environment that can be modulated as compared to the tissue, the interstitial tissue in the subcutaneous space. Therefore, the combination of the chemistry that he can afford using different type of hydrophobic, hydrophilic molecule in combination, and the protected environment offered by the implant really offers a spectrum of tunability that cannot be done, cannot be achieved otherwise. So we are working jointly since a couple of years ago on to the development of a platform, MPT platform, that combines the polymeric chemistry technology from his laboratory and my nanofluidic implant technologies from my lab. And we are targeting a product that we at least feel that may have some level of acceptability. Of course, we understand that the end user research is needed, and we are interested in performing that as soon as possible. But again, the ideal dose of etonogestrel that we are targeting is about 35 microgram per day. We started working with islatravir, as I showed you earlier. However, of course, there is the news, the fact that the Merck has interrupted the development of their own implant. which is to a certain degree is not a good news in the context of the development of islatravir for HIV prevention. However, there are analog of islatravir that have been developed by Merck, and there are other antiretroviral drugs that we can study in combination with tonogestro for the MPT. And we are in the process, in fact, to transition from this antiretroviral to others. So next slide. So in conclusion, we have a control release technology that allow us for the scalability, can be scaled in terms of microfabrication method, can be fabricated in large number with low cost. So therefore has relevance not only in the developed countries, but also in the country under development. We have a control zero order kinetics that really avoids any fluctuation and burst or decay, release, and PK tail and terminal dose dumping, which often can be observed in some of the injectable product and polymeric drug delivery systems. So this technology really enables ultra long-acting, multi-year delivery, where we don't need to worry a decaying profile that may fall underneath a preventative level. Also, we have the capability of doing transcutaneous refilling of solid drugs, and therefore an implant that may be inserted and deliver drugs for about 2-3 years continuously can be then refilled with a very minimal invasive procedure in a doctor office, and therefore continue for other 3 years without major complication. And finally, the technology we like to call a platform because offer broad applicability and versatility. We have tested in the context of various antiretroviral drugs, including tenofovir, lefenamide, and tricytabine, cabotegravir, bictegravir, of course, islatravir, as I've shown, and also combination of those. So we believe that there is the possibility to use it in the context of other type of application beyond HIV, in other infectious diseases, perhaps in other MPT type of technologies. Next slide. Finally, I just want to disclose that intellectual property developed in my laboratory has been licensed by the startup company called Continuity Biosciences. Next slide. And together with the company, we have started the process utilizing NAMSA, which is a CRO that has been helpful in determining the path forward toward clinical translation. We have determined some of the step required for us to move into clinical trials and how long it would take, depending, of course, and what is the amount of funding that would be required. So I can say that the path to clinical trial, first in human clinical trial, is not exceedingly long, but of course it depends on the success that my laboratory as well as the company will have in raising the proper funding. Next slide. With this, I would like to thank you for the attention. I would like to acknowledge my group and the funding sources. Of course, we have received funding from NIAID. 2 grants are supporting this type of work. NIGMS as well has supported us in the past for the development of the membrane technology. And we're working closely with other in the industry in the field, including Gilead. Hopefully, we'll be able to work with many other groups in collaboration. And of course, I acknowledge all the collaborators, including Patrick Stayton, who will present just right after me right now. Thank you.
Great. Thank you so much. We're a little bit behind, so I just want to very quickly introduce Dr. Patrick Stayton and ask everyone if you have questions, you can drop them into the chat or hold them for the end. So thank you so much, and we'll turn it over to Dr. Stayton.
All right. Good morning, everybody. It's a pleasure for me to be for the first time with this group and present to you earlier stage, I would say, technology, but I'm looking forward to, especially to any connections that might come out of this presentation. As Alessandro alluded, we've been developing a new platform to control the release of small molecules. And it actually was developed initially with the Bill and Melinda Gates Foundation. And so our target profile, product profile is one that you would all be familiar with. It's really to extend as an injectable depot from the current Apretude 2-month time period upwards to probably about a year as our initial target. But to have a better control over the PK. And as Alessandro also alluded, if you really look at the current injectable products, they still suffer from initial burst release or maybe higher than desired initial release, and also end-stage release that's not ideal in PK tailing. So our goal was to develop a new platform to address those key PK shortcomings. We also, though, wanted to see if we could lower injection volumes so that they are more comfortable for women to use, and to develop a sub-Q product compared to an intramuscular product, again, to make it more acceptable for patient use. What comes along with that is maybe the hope that we could have more of a self-injectable system, a reduced clinic and healthcare worker requirement. And right up front, it was quite a constraint for us to think about this problem for the first time with the reasonable cost constraints and limited cold chain. And then we were also tasked with the idea of could we develop a platform that might deliver different defined time periods, as I'll allude to you. Now, although this was developed as an injectable system, at the end of the day, these polymeric prodrugs can be readily adapted into implant devices, such as Alessandro's beautiful devices. They're polymers. They can also be blended into things like intravaginal rings. And so they do offer other alternatives as part of devices and implants. The concept, I'm going to show you some chemistry. So I know I'm trying to do this chemistry light for everybody, but at the end of the day, it really is a chemistry-based platform. And what we're doing is synthetically making polymerizable monomers that have a defined linker that controls drug release. And these are called a prodrug monomer. And then these can be polymerized then into a final polymer. And sometimes we do a copolymer, and that tunes the properties, for example, the hydrophobicity or lipophilicity of the resulting polymer. And it turns out that it can also help. It's a control knob to tune the release kinetics. So at the end of the day, we make these polymeric prodrugs. And what's new about this as a depot from the types that I even teach about in my class is that there's really nothing else involved. And so there's no other excipients. The polymeric prodrugs are put into an injection solvent like DMSO and injected, and they gel and form a depot directly. So at the end of the day, the depot is just the polymeric prodrug in an injection solvent. So the data I'll show is from my own group in bioengineering at UW. Sam Arnold's group here at UW did a lot of the PK modeling that I'll show you with islatravir. And then we also did some of the chemistry with a group in Australia at CSIRO. So we've published the initial work with this system using the Gilead drug TAF. And I'll show you mainly results with islatravir now, the Merck drug that Alessandro also mentioned. And these we found, have a role in the properties, but in general, this platform works super well with different drugs. So there is a sophisticated drug space that's available, and the synthetic chemistry behind this is really key, I think, to its adaptability, as I'll show you. So at the end of the day, these are designed to be sub-Q, low volume, viscosity, narrow gauge needle injections for women to find better than the current product, which is intramuscular in 3 mils. Zero order release is always our goal. It's been hard to achieve with injectable systems. And I'll mention and come back to the low cost of goods. The mechanism for these, I'll just show in cartoon fashion here. Up above, you can actually see by IVUS imaging, if we fluorescently label the polymer chain, you can see this nice gel that's formed. And what's happening is it's actually the hydrophobicity of the drug in the polymer chains that drives a gelation, and it forms a very tight injectable depot. They also have to have a mechanism for clearing over time. And rather than like a biodegradability of PLGA, it's actually the degradability that is controlled by the linker, the prodrug design. And as those drugs are released, those positions become carboxylated. And that negative charge that's developed in this low dielectric environment of the gel causes a swelling. And at the end of the day, it's actually the dissolution of the gel that drives the termination phase or the shutoff of the depot. And so it's an interesting new depot. The degelling and depot shutoff are directly connected in this case to drug release. So there's a lot of knobs. I don't have time to show you them all. Some easy ones to understand, I think, just conceptually are, if we make polymers with different linkers that have intrinsically higher hydrolytic rates, higher activation barriers to hydrolysis, like an alkyl ester compared to a faster releasing benzylic based linker. In the red and the orange, you can see that the benzylic linker releases faster. It shuts off after about 90 days. The alkyl linker releases lower over this time period and didn't really have such a great profile. So we've kind of focused then on the benzylic group. But then over on the right, you also then can increase the amount of the drug monomer, which is a positive because a higher percent of your mass is the drug. And there we get in rat, very nice profile of over 7 months of pretty close to zero order release and very good shutoff. So these are depots that are engineerable at the level of the polymer. And I find that kind of fascinating that you can engineer the polymer and the monomer in very straightforward and intuitive ways and have such a nice impact on the bulk depot properties. There are many other knobs to turn. A simple one is molecular weight. So as you go from about 8,000 molecular weight, you double it to 16, and then double it again to around 30,000 molecular weight. So these are actually relatively low molecular weight polymer chains. You can see the extension of time where the higher molecular weight has this very nice profile, again, out to many months. And if you look at the release per day, you can also see, I think, from Sam Arnold's modeling of this work, that the nice shutoff as well. So the higher molecular weights, the right copolymer, and these can have really excellent release properties. The other thing I'd point out is these are only 200 microliter injections. And so there's a lot of upward space in terms of the amount we would deliver and the duration, but these are already, out to a time period that is attractive in humans in a very small volume. And the key to that, again, is that high drug weight percent. Most of the mass that's being injected is the drug. There are many other knobs to turn. At the monomer level, you can push the linker out to make it more accessible. With this spacer unit that is ethylene oxide spacer, go from 1 to 4 to 7. We did this with the Australian collaborators. And you can see that the EG1 actually is very stable. That gel doesn't release drug, even though it's a non-covalent gel, that's very stable. Too stable to really release. As you go to EG4, though, you get a nice release profile. Again, this is only 200 microliters injection. And so this could be driven upwards, which is higher volumes. And then the EG7 is a higher daily release, but then shuts off earlier at around 120 days. However, it's really remarkable. The EG4 releases as an injectable system for almost a year time period with a nice, pretty zero order release profile. So especially for contraceptives, for example, that might be already getting near an interesting daily release profile. Finally, with Alessandro, we've been so excited to work with his group, and it's been very productive for us. We did an initial non-human primate study. This is just a pilot N=1 that we're going forward with now. But these profiles have held up in the non-human primate. Very little uncontrolled early release, very nice shutoff. And so, again, a very relatively low volume of injection that could go upwards still. The cost of goods is something that, I've learned is very important in this space. And it was something that drove us for this, simplicity of just having a polymeric prodrug as the only excipient. It's not really a true formulation in a sense. And the Gates Foundation did a nice manufacturability and cost of goods analysis with us. It was done by George Tyson, who is probably the person best positioned to do this in the world. He's brought some of these types of polymers in industry to the market, and then also he teaches this at Berkeley. And I was excited we're able to publish this. It's not a normal part of many papers, but it was found to be commercially manufacturable at existing reactor scale up to 50 metric tons. That's a true global scale of this kind of polymer with existing India, China capabilities for both the monomers and the polymers and at GMP. And there's a scalability to the cost, of course, but these are good, I think, markers at this stage. Less than $1 per gram at the higher scale, and as we went down, still less than $3 per gram, which is probably an overestimate per dose. And so these appear to have a manufacturability and cost of goods sold footprint that is favorable for use, around the world in some disadvantaged settings. So at the end of the day, if you compare the release profile, I think to cabotegravir plasma PK that's been published, you see the stark difference in these kind of zero order release profiles as an injectable to this liquid formulation. And so that is what our hope is this will hold up as we further develop these. But the low volume in the sub-Q compartment could provide advantages as well. We have gone on to make etonogestrel with Alessandro for our MPT project. So these can be adapted to different drugs. And with etonogestrel, we're only the first 30 days, but it appears to have a very nice profile. And we started this with only a 50 microliter injection. So there's a lot of room upward here to go. But that's really its advantage. And we're also excited to think about these, especially for lipophilic drugs, as a way of tuning release and changing their solubility properties in that protected environment of implants, or as blends into other types of devices. It's a way, I think, of gaining control over the small molecule release in a variety of different platforms. So thanks once again for the chance to present today, and look forward to any questions that people might have.
Great, thank you so much. We're going to pivot now and turn to Dr. Taratula, who's going to talk a little bit about his research that's less directly immediately applicable to MPTs right now, but potentially something to explore in the future.
Can I get the chance to share my slides? Dr. Stayton, can you
I'm trying.
Allow me to do that.
I finally got the click, sorry.
That's perfectly fine. Right. Good morning, everyone. First of all, I would like to thank the organizer of this webinar for the invitation. It's my great pleasure to be here. And as was mentioned by Clara, so my lab use nanomedicine for the treatment and diagnostic of different diseases. And today specifically, I would like to discuss one of the projects in my lab in collaboration with the Oregon National Research Primate Center, and specifically with Dr. Oleh Taratula. And this project is focused on the development of novel therapy named magnetic hyperthermia for the treatment of endometriosis. For those who don't know about this disease, I just want to mention here that endometriosis is the disease associated with the female reproductive organs. And briefly, endometrium is the lining of the uterus. And what happened in the endometriosis, according to the Sampson hypothesis, those endometrium cells, they can pass or retrograde through the fallopian tubes during menstruation and sit in the peritoneal cavity. So basically, they form the lesion in the cavity. So this disease can affect more than 170 million patients worldwide. And it's very nasty disease, is associated with infertility and severe pelvic pain. Unfortunately, these days, we currently don't have a cure for endometriosis. The available medical therapies which we have, those hormonal therapies, they rather prevent the symptoms such as pelvic pain, but they don't eradicate endometriotic lesions. And patients who want to improve their fertility, they often go for the surgery, but unfortunately, the surgery, recurrence of the disease after the surgery is 50% or even more, and patients very often, they need like multiple surgery. Well, so our goal was, when we started this project, our goal was just to develop non-invasive therapy for endometriosis treatment. And our choice was magnetic hyperthermia. And what I just want to mention, magnetic hyperthermia, this is the experimental treatment for cancer. And it's based on the concept that magnetic nanoparticles, when you deliver them to the cancer tumors, and after that, you expose the part of the body to the magnetic field. Those magnetic nanoparticles, they can absorb the magnetic field energy and generate heat. And as a result, they can destroy cancer tumors. And right here, you can see the medical device, which is currently used in clinical trials for the delivery of alternating magnetic field. So unfortunately, magnetic hyperthermia, which we use for cancer, has a number of limitations, and one of them that this therapy can only be used for the treatment of localized and accessible tumors. And one of the reasons for that, magnetic nanoparticles and any nanoparticles, when you inject them systemically, only the small percentage of the injected dose can accumulate in the cancer tumors. And on the top of that, the currently used magnetic nanoparticles, they have moderate heating efficiency. And what happened, those nanoparticles in current practice, they deliver to the tumor after intratumoral injections. Obviously, it was not a choice for us because endometriosis is, in case of the endometriosis, lesions, they can be spread in the abdominal cavity. And the only choice for us was systemic administration of nanoparticles. Well, when we started this project back in 2017, so there was little known about the application of nanoparticles for endometriosis. And we we were not sure if we inject those nanoparticles, they will accumulate in endometriosis lesions. So we just decided just to take some knowledges from the cancer field, and specifically what we know in the cancer field, that after systemic administration, nanoparticles, they can accumulate in the cancer tumors via passive targeting, or just briefly, when the nanoparticles, they reach the tumor, and the tumor has leaky blood vasculature. So nanoparticles, they can escape from those blood vessels and just stay in the tumor. And we also know about the active targeting, and active targeting is related to the fact that cancer cells, for example, they can overexpress many receptors, and if you modify the nanoparticles with ligands that target those receptors, so those nanoparticles, they can internalize in those cells and just stay there for some period of time. Well, so first of all, we decided to see if it works, and our major hypothesis was that endometriosis is also angiogenesis-dependent disease. And we also knew from the literature that endometriosis also has leaky blood vasculature, and we decided just to test if nanoparticles could be delivered to endometriosis lesion after systemic administration. So endometriosis happen only in human and non-human primates, but my collaborator, Dr. Stayton, he can develop animal models of endometriosis. And our first step was we use his mouse model with grafted endometriosis lesion. And what we noticed that when we injected polymeric nanoparticles labeled with near-infrared fluorescence dye, so we saw that our nanoparticles, they can accumulate in those lesions, as you can see from this signal in those lesions. And on the top of that, when we did the histology, we saw that those nanoparticles, they also can escape from the blood vessel, and blood vessel here represented with this yellow color, and the red color shows our nanoparticles. Afterwards, we also confirm this accumulation of systemically injected nanoparticles in endometriosis lesions by using rhesus macaques with grafted lesions, and you also can see here that our nanoparticles, they can go to those lesions. So the next step obviously was to develop magnetic nanoparticles with high heating efficiency, meaning the nanoparticles which can generate a lot of heat when we expose them to the alternating magnetic field. And probably it was the most challenging step for us, but our lab was able to develop a method for the preparation of iron oxide nanoparticles, and specifically we were able to modify the shape and morphology. And here you can see the TM image of our nanoparticles. So we developed hexagonal shaped nanoparticles, and those nanoparticles, they also have a core shell structure. And what we noticed that these nanoparticles, they have at least 10 times higher heating efficiency than conventional spherical nanoparticles, which currently used for the magnetic hyperthermia. Well, to make these nanoparticles targeted to endometriosis cells, so we analyzed different receptors which overexpress on those endometriosis cells. And here you can see the staining of endometriosis tissue, which we collected from rhesus macaque. And we noticed that the KDR receptor is highly overexpressed in the endometriosis lesions. So to make these nanoparticles targeted, our iron oxide nanoparticles targeted, we encapsulated them in the polymeric nanoparticles, and specifically by using the biocompatible polymer called PEG-PCl. So what was done, we conjugated the KDR peptide targeted to that receptor, and we end up with this nanoparticle. As you can see here in this image, we have our iron oxide magnetic nanoparticles, and those nanoparticles, they are covered with the layer of the polymer and that polymer has the targeting peptide. Mouse studies just show us that systemically injected those nanoparticles after intravenous injection, they can accumulate in the lesions, efficiently in the lesions. And also, I just wanted to mention that our iron oxide nanoparticles, they are very good T2-weighted MRI contrast agents. And actually, it's a benefit because what we can do when we inject nanoparticles, we can, by using MRI, we can identify if they went to the lesions, and only after that we can apply the magnetic field. And what you can see here, we have the loss of the signal when the animals were treated with our nanoparticles. And finally, we collected this tissue and stained them for iron just to detect if the iron oxide nanoparticles are there. And you can see here this blue color just represent iron oxide nanoparticles, or iron specifically here in this case. We also evaluated different organs, and what we noticed that our nanoparticles, they can efficiently clear from other organs, and we only can saw only some accumulation of them in the spleen and in the liver. Interest was very interesting when we treated mice with those nanoparticles, and after that, when we apply external alternating magnetic field, we were able to increase the interlesional temperature above 50 degrees. And right here, you can see the result of the therapeutic outcome of this magnetic hyperthermia. So here's the mouse which was injected with our nanoparticles, and 5 days later, we exposed that mouse to the alternating magnetic field for 20 minutes, and we were able to eradicate those endometriosis lesions, and we didn't see any recurrence, for the over 50 days. We also show that this therapy, we can selectively apply alternating magnetic field, and here you can see another mouse with 2 lesions, and only this lesion was treated with alternating magnetic field. And as you can see here, we were able just to eradicate that lesion. Obviously, we did safety studies, and we showed that mice, they did after the treatments, they don't lose body weight. We did the blood analysis, and we didn't see any toxicity. In the next step, we moved to rhesus macaques, and specifically this time, we were able to identify animals with spontaneous endometriosis. And the major question was if nanoparticle, they also can accumulate as efficiently as in grafted lesions. And here you can see the MRI image of rhesus macaque. And this is the image before injection of nanoparticle, this one after injection nanoparticles. And we can see in this lesion, we have the loss of MRI signal, just indicated that our nanoparticles also accumulate in spontaneous lesions. We also did the analysis of lesions and different organs, and we notice very good accumulation of our nanoparticles inside of those lesions, and was little accumulation in the liver and some other organs. And finally, where we are right now, we also tested if we can generate high temperature inside of this lesion. And what we can see so far, so we can raise the temperature of the lesion up around like 46 degrees. But I just wanted to mention here that the dose which we use in rhesus macaques was 2 times lower than in monkeys, and we still have a chance to increase the dose. So our next step would be we are about to start the evaluation of therapeutic efficacy of this magnetic hyperthermia in macaques with spontaneous endometriosis. And I hope soon I can share more result. And finally, I would like to acknowledge my lab members, my collaborators, specifically Dr. Oleh Taratula, and finding agencies, including NICHD, for the financial support. Thank you for your attention.
Great, thank you so much. I feel like we're already in the future with your presentation. And so our last speaker is going to be Dr. Kevin McHugh, and then we're hoping to still have a little bit of time for Q&A afterwards. So without any further delay, Dr. McHugh.
Okay, great. Yeah, thanks so much. So hard to follow those presentations, but I'll do my best here. So today I'm going to be talking about this system that we've developed based on peptide hydrogels that allows to deliver drugs over long periods of time in a few different classes. My lab is specifically interested in injectable systems that are inexpensive to produce and use. My background is kind of in thinking about vaccine delivery for low resource settings. So hopefully the things that we make are broadly useful to people regardless of their kind of income level. And so, I won't belabor the background here because I'm sure everyone knows it a lot, but, drugs are great. They help a lot of people. They extend lifetimes. They improve quality of life. Many people take drugs. Half the world's population takes at least one drug a day. And if you're over 60 in the US, that's 85% likely that you take at least a drug a day. And so unfortunately, drugs aren't perfect, right? So they're not perfectly effective. They have side effects associated with them, and we have low patient compliance, which is actually maybe an underappreciated issue by the public. If low patient compliance was a disease in its own right, it would be the seventh cause of death in the U.S. Seventh leading cause of death in the U.S. And so, we in my group are not drug developers. We don't make new drugs. We think about how we can overcome some of these challenges. Specifically, how can we make these drugs more effective, reduce their toxicity, make them easier, more convenient, cheaper to access. And so today I'll be talking about a hydrogel system. So hydrogel looks something like this. So, you can think of kind of Jell-O, if you will. They have different properties, but in particular, they form these networks right at the nanoscale, and then they look like these gels that you see on the left in the kind of macro scale. And so we can use these to control delivery over long periods of time, like Patrick was talking about earlier. And we can also potentially use them to create local concentration gradients, like Alessandro was talking about previously. And so we're specifically interested in using peptide hydrogels. So these self-assemble. These are peptides that can be engineered, designed basically based on their amino acid sequence to assemble. Okay, and if you look at them, they look something like this. We're particularly interested in this class called multi-domain peptides, or MDPs. This has been worked on by our collaborator, Jeff Harkrink, in the chemistry department here at Rice University. And so the idea is that you can have this kind of middle alternating sequence of these serine leucides. And basically what that does is make one side hydrophilic, one side hydrophobic. And so those hydrophobic sides collapse upon each other. You need these charge groups at the end to make them form fibers instead of something bigger like a sheet. But nevertheless, these can self-assemble to form these structures. And we have some choice over what kind of amino acids we put in this, depending on if we wanted to, say, release a particular drug that has a charge slower or faster, or we want it to be more inflammatory, say for a vaccine, or maybe less inflammatory for some type of drug delivery for a chronic disease. And so these MDPs can be really inexpensively produced using solid phase peptide synthesis, a very well understood technique that won the Nobel Prize 40 years ago or so. So very inexpensive to do this, very easy. I have lots of undergrads in my lab doing this on the daily, so not difficult. The other thing we like about MDPs is that they're composed of these amino acids, which are naturally occurring, so they can fully degrade into their constituent components, and they're very well tolerated in the body, so we don't see a large foreign body response. These fibers are held together by intermolecular forces. So they are not covalently cross-linked into one big molecule, essentially. They are held together by those hydrophilic sandwiches that I was talking about before. And one interesting property that this confers is that it makes them injectable. So they're shear thinning. What that means is that you have a gel if you're not pushing on it, but then when you push it through a needle, it flows like a liquid, and then it rapidly self-heals on the other side, forming a gel again. So this is really convenient from administration perspective because we don't have to necessarily do something that's more invasive. We can just directly inject this. And then because it's an aqueous formulation, we don't have to worry about exposure to organic solvents or high heat or anything like that that could denature certain drugs, in particular biologics, which I'll talk about at the end of this presentation. And so one issue, which is pretty challenging for the drug delivery space that it doesn't usually hold on to drugs that long, in particular small molecules. And so if you look on the right, this is a hydrogel, this MDP hydrogel shown. Shrink this. In blue, it releases over a handful of hours, right? So that's not necessarily useful for a ton of applications. And so we were interested in thinking about how we can improve the interactions, increase interactions between the hydrogel and the drug to make it hang on for a lot longer. And so the one we're particularly interested in using is dynamic covalent bonding. And the way that you can think about this is that there are two states of these molecules, one in a bound state where these two pieces are together, covalently bound, so it's a strong bond, and then another state at equilibrium where some of them are not together. So there's two separate parts. And the one we're particularly interested in using is this dynamic covalent bonding between boronic acids, which is shown on the bottom left here, and these diols. And so the way to think about this is that some are in each of these states at all times, and they're going actively back and forth, right? So it's a dynamic system. And so we thought, well, maybe we can put one side, the diol, on our hydrogel, and we can take the boronic acid containing drug and mix it in there, right? And so instead of just directly diffusing out very quickly, some of those molecules are going to engage in this covalent bonding with the hydrogel, and that is going to prevent diffusion while it is bound, right? And so the idea was that we could stretch out the timeline over which these drugs that have boronic acids would release. And so we made a library of these MDP modified hydrogels, right? So we can put these different diols on there. The one that's been used a little bit in the field, not so much with peptides, are these catechols shown in the top left. There's some things not to like about those, like oxidation potential. And so we're really interested in these SHA modifications, which we'll just underline the word here. But think about SHA. So that's the one we thought would work the best based on our understanding of the chemistry. And so good things that this modification on the bottom left shows that it doesn't change the structure that's formed. It's still able to form these beta sheets. It doesn't change the hydrogel properties of it, and it's still rapidly self-healing. So all the things we liked about MDPs, we still like about these modified MDP hydrogels. And so then we went and started with these small molecule drugs that contain boronic acid. There's a number of FDA-approved ones, lots more in development. This turns out to be a useful group for medicinal chemistry, so we're getting more and more drugs that have boronic acids on them. And so first we did these delivery or release experiments in vitro, and so these are all small molecule drugs that contain this boronic acid. And so really the key here to look at is the black line on the top. So that's if we don't have any dynamic covalent bonding. And so we're reaching equilibrium very quickly. All the drug is basically coming out into the surrounding solution, or at least reaching this equilibrium, right? And that happens very quickly. But if we put on our SHA modified hydrogel, we can really stretch this out considerably longer for each of these different drugs, because we are basically tying up some of that drug covalently onto the hydrogel. So that was exciting for us, and we wanted to then investigate some in vivo models. And so we took bortezomib, which is this multiple myeloma drug, so a cancer. And so people don't use this for local tumors, solid tumors. They use it for only this liquid tumor. And so we wanted to investigate, can we get more drug in a particular location? So maybe make this drug, which is not suitable for solid tumors, into one that is suitable for solid tumors. And so our goals are really to increase the concentration of the drug inside at the injection site while decreasing the systemic amount of drug that would otherwise just cause kind of side effects. And so here's what we see in the blood concentration side. So if you just inject the soluble drug shown in black, you get super high concentration. This is going to be associated with toxicity. If you put that same amount of drug into a hydrogel, our SHA functionalized hydrogel, we can keep the peak levels very low and have them persist for a long period of time, right? So we potentially decreased our systemic toxicity of this particular drug. And on the other side, if we look at where the drug is actually injected, so we can take a look weeks later. This is mass spec imaging. So basically this is telling us how much drug is in the tissue right in the surrounding area after several weeks. And so if we inject just the regular soluble drug, and we have other groups here as well, you don't really see much left after 3 weeks, right? And that's expected. It's already diffused out, it's dissipated, it's been cleared, as you can see from this line in the graph. But if we take our SHA functionalized hydrogel, now we see a persistent high concentration of our drug at the injection site. So that was exciting for us. So what do we want to use this systemically though, right? So another one of these drugs that's boronic acid containing small molecule is for tuberculosis. TB has really bad adherence issues. Lots of people will get prescribed, weeks or months of drugs, take it for a few days or a few weeks, feel better, and then walk around with latent TB that they can spread for the rest of their life, or maybe even induce, drug resistance. So we would love to have a solution that is potentially has no potential for errors, right? And so GSK is one of these drugs that's been developed by, as you might have guessed, GSK. So it's GSK 656. It's a small molecule drug. It has this benzoperoxyl group on there, so we can play our same game with having this dynamic covalent bonding to our hydrogel. And so we did a pharmacokinetic study. And so if we just take the drug, and that's in black, and inject it, we get a high peak concentration, and then we drop below this effective dose 50 within 3 days, right? So not a very good strategy to just load up on a bunch of drug and have it rapidly get cleared, right? But if we put it in our hydrogel, which is shown in this kind of magenta color, same amount of drug, but now it's inside that hydrogel that it can dynamically covalently bond with. Now it persists for over 3 weeks inside there as it drops off slowly and maintains its efficacy. And so then we worked with Eric Nuremberger at Johns Hopkins University, who has infection models for TB, and we showed that we can basically suppress the growth of an infection using this gel, right? So if we just take a gel that's empty, basically an untreated mouse, we see rapid increase in the loads of bacteria in the lung. However, and if we do oral dosing, which is the kind of current standard of care, we do get a little bit of an increase. So the daily oral dosing. But if we use our gel with the same amount of drug and release it, then we get basically no growth in the bacteria in the lung. So that's really exciting for us. And we're looking at ways to stretch this out, but it seems very promising. And so not every drug has a boronic acid on it. So we asked ourselves, can we add a boronic acid to some small molecule drugs that don't already have one without affecting its function? And so we use this toll-like receptor 7 agonist, which can be used as basically an immune modulator. And we added a phenylboronic acid to it. That's what the chemistry looks like, pretty easy overall. And we saw that it doesn't affect its ability to activate the toll-like receptor 7 surface protein. And if we put it in a hydrogel that allows for dynamic covalent bonding, it does get released over a long period of time. So that was exciting for us, and we wanted to keep pushing farther. So then we moved into thinking about biologics. And so as you may know, diabetes is a huge issue. And so people self-dose insulin in response to meals, but also this kind of basal insulin that people can administer once or twice a day that kind of just provides some correction against hyperglycemia. And so we thought maybe we could make the system or use the system to deliver insulin in such a way. And so we have the site-specific modification of insulin with, again, phenylboronic acid. So we're putting a boronic acid onto something that doesn't natively have one. And then we investigated its release in hydrogels. And as you can see, when it has the ability to undergo dynamic covalent bonding, it comes off much slower. It's not very sensitive to glucose. So actually one way that people have used the catechol version of ours, so we're using SHA as our modification. If you use the catechol version, it's very glucose responsive. So people actually use it as a responsive tool. We want it to be non-responsive, right? It would be really bad if your drug released a little bit more when you had a big meal or something like that, right? So it's really relatively non-responsive. And we showed that we could use this system to correct insulin level or correct blood glucose levels in diabetic mice for up to 6 days, which is obviously much better than having to redose every 1 or 2 days. If you just inject a regular amount of insulin, you take your high blood glucose, right? Because these are diabetic mice, you rapidly crash down into the safe level, but in the matter of an hour or two, you're back into this hyperglycemic levels, as opposed to in our case where we have control release version, we can stay in this good level for stay in this good range for a long period of time. So that was really exciting for us. And then right now we're really interested in adapting this system even further to use them for antibodies. So obviously antibodies useful in a lot of spaces. And so they have we can use the same chemistry, put on phenylboronic acid onto these proteins to allow them to dynamically covalently bond, basically stick to our hydrogel for a little bit longer. And so we've shown things like that we don't reduce the activity of these antibodies by very much, even if we don't use site-specific labeling, using a couple of these clinically relevant cancer antibodies. We show that they stay stable in the gel, and then we show that we can get them to release over long periods of time. So this is 8 weeks in vivo. So if you just take the soluble antibody, inject it subcutaneously, it's gone by a couple of days. If you put it in the gel, then it's around for 8 weeks, which is basically when the hydrogels degrade. And so really we're thinking about pushing this farther by making some slower degrading hydrogel bases. So in summary, we created this library of these diol-containing hydrogels, right? So they're self-assembling, so we can inject them. They're self-healing, and they're shear thinning, so we can inject them. We can make them very cheaply. They can really slow down the release of a bunch of these drugs. We can use that for local delivery or for time delivery or some combination of the two. And we can do this with small molecule drugs that already have boronic acid, small molecule drugs that we add a boronic acid onto, or proteins that we add a boronic acid to. And so really we're looking to expand the kind of drugs that are compatible with this. So it's pretty trivial for us to employ the same approach we employed for antibodies and insulin for other proteins of interest, therapeutic proteins or prophylactic proteins of interest, and then thinking about other ways that we can optimize the hydrogel matrix so that we can really push out beyond the 8 weeks. And we have some ideas on things we can do there, and some hydrogels that we've seen stick around for at least 3 or 4 months to push that out. So with that, just like to thank my team. This work was led by recently graduated PhD student Brett Pagostin, and a couple of undergrads that were working with him. And then I acknowledge Jeff Harkerink, who worked with the MDPs, Zach Ball, who worked with SHA, and a few other folks as well. So thank you all for listening, and happy to take questions during the discussion portion of this seminar. Thanks.
Great. Thank you so much to all of our speakers. We're going to have a short Q&A, maybe about a little less than 15 minutes. That's how much time we have, but I think everyone's presentation was so interesting and compelling that we didn't want to hurry anyone too much. And it's also some really interesting and in-depth science that's happening, so it's not easy to do quickly. So actually we had already a question about tuberculosis. So I'm going to say, Dr. McHugh, you've already answered that question. So thank you for that. That was from one of the participants, just asking if anyone had been investigating applicability to TB. And so I wanted to, we've gotten some questions in the chat that Bethany will also lob, but I wanted to also ask, we had a question from a participant about sort of community settings and sort of what settings that your research might be applicable in terms of like clinical settings. Are they things that need to be done in a facility? Can they be administered by community settings? Or for example, for some of the implants, can they be refilled by a nurse or a pharmacist even? So yeah, I don't know if any of the speakers want to talk to the level of professional and provider who's going to be administering these interventions.
So perhaps I can start with that question. So we have done a little bit of a research interrogating various different clinicians, and show the product that we are developing and ask for their feedback regarding the potential deployment, both in a setting where there are abundant medical resources and infrastructures, such as in the US, or in other setting where perhaps those are lacking. And we understood that the surgical procedure that is required for an implantable device is minor. So that can still be performed in a doctor's office under local anesthesia. It doesn't require, obviously, a very, invasive procedure. And at the same time, the post-operative follow-up and checkups, it's still minimal. And anything that has to do with the transcutaneous refillability of these devices may not even require a clinician's input. In fact, it may be done, for example, by a nurse that is trained for the procedure itself. And part of the process that we have used for developing this platform is really making sure that the procedure itself is simple enough and straightforward that really mitigates risk associated with the actual manual administration of the drugs. We have also started looking into developing of tools such as injectors that can be used and multiple uses, or they can be disposed upon use, and so really to simplify this procedure. Ultimately, we need to go through a rigorous end user study, and perhaps also a little bit better in-depth analysis of what would be the clinical practice. But this first assessment indicates that we may be able to do this in various settings, including those that have limited resources.
Yeah, just speaking about my technology, I mean, I think the idea is that, yeah, we can have any type of healthcare provider should be able to do an intramuscular injection. And then, I think there's possibly the opportunity for self-injections depending on the application and if it's okay to, say, inject in the IP space or something like that, right? So people do, we inject our gels with insulin needles already. So I think if people can give themselves insulin, they probably have the wherewithal to do this self-administration. Of course, it probably depends on if this is a chronic disease and if it's worth training someone, or if it's just a one-time injection that it will cover your whatever treatment period and then you're okay. But yeah, I think the idea of making these this way is that they're super easy to implement for everybody, so that's not a barrier to use.
Maybe are there any other responses to that? Otherwise, I'd like to read a question to Dr. McHugh, but I don't want to cut anyone off. Okay. So this is from Frederick Nitko for Dr. McHugh. Have you considered therapeutic molecules for the treatment of Hep C? GSK's developed a group of NS I can't read it, RNA polymerase inhibitors that contain boronic acids.
Yeah, so we have not, that's not for anything intentional. I would say it seems very likely that it's would work similarly to the other small molecules that contain boronic acid. So I would, this is actually the results that I was presenting are not published yet. So we're really just starting to develop this system. And that's why we haven't quite turned all the knobs that we can, right? So I have I mentioned my graduate student just finished his PhD. The new one who's taking over is already kind of thinking about how do we move beyond what we have already? Can we improve these things and make them last even longer? So I think that's definitely of interest to us. I think it seems just a brief look, it seems like those are pretty potent drugs, so we would be able to use those with us, and it would just be a matter of studying it and having a good animal model. So we are not experts in all the animal models. That's kind of the nature of being kind of these platform builders, is that we can't be an expert everywhere. So if we have a good partner like we did in the case of, say, TB, for example, that has a good animal model that we could test efficacy in, then I think that would become very interesting to us. So that maybe those are good drugs to follow up with. Thanks.
Yeah, that was a great question. Thank you. Are there any other questions for the chat? Otherwise, I'm going to pass it over, Claire, if there's any. I know we have a lot of other questions that we were interested in, but I just want to say this has been fantastic. Really, thank you all. So, Clara, anything else? Otherwise, we'll wrap up.
So I wanted to tie it back, and we had a couple people start mentioning end user research. And so one of the last webinars we had was looking at social behavioral research and end user research. And I wanted to know if you started incorporating any of that into your own research, and at what point you would start doing that. And if you have an understanding of how acceptable and interested people are in some of the technologies that you're researching.
Well, I could say a few words about the injectable space, and it of course isn't my own research into user assessments, but right from the beginning, we were given some constraints around injectables, and they were technical in the sense of things like what our target duration should be. For example, 1-year product based on what's coming down the clinical pipeline. But it gets back a little bit to the first question too. For injectables, the device side of injectables is really expanding, so fast in the pharma field. I work in general in that area. And what does that mean for this space? It's kind of an inequity that self-injectables haven't been the focus, of course, of the main pharma world. But the idea that women in sub-Saharan Africa could self-inject, so not even need a clinical setting, is one that I found fascinating and very important concept. So when you move to sub-Q, so many people in our worlds are self-injecting, and I self-inject an antibody myself regularly. the volumes now are up to 2 mils. For example, for the antibody that I inject, it's a 2 mil self-injection. And the device advances there are really fantastic. But what I haven't seen connect in really is this idea that how would women in sub-Saharan Africa feel about self-injecting, and how would that fit into their world using these new injectable devices, some of which now, are prototyping at up to 10 mils. And I think that's an area, it's not really answering the question, it's answering the question with a question, but I really feel like that space industrially, because of the push for antibodies and self-injecting antibodies outside of our clinic setting, has driven that. And it seems very doable to me to think about it, but then there's the cost element. And what does the device cost? What's an acceptable device cost? What's the cultural or user setting for self-injecting these kind of products? I think that's a really important area for all of us to kind of get a grasp on, because there's such advances from that device side from pharma, and such approval validation of these, that I really am thinking a lot about that space. And it pushed us to go sub-Q right from the start with our platform, with the idea that it could get outside for users of even a clinical setting. So I'm not sure that's really an answer, but maybe it also has some important questions for us to consider as a field.
Thanks so much. I really appreciate that you're starting to think about those kinds of things so early on. So we have just a couple minutes left, so I'm going to let Bethany wrap up our webinar.
Yeah, I just want to say, I think we could have talked about this for another, like, twice as much time. So thank you again, really fascinating. For those that are here, I want to just remind you that all of our webinars, including this one, is recorded. And so the recording and the slides will be posted shortly. There might be a few modifications to the slides, with after the presenters get them back to us, just with data that is comfortable to share. And I encourage you to also visit the IMPT website. There's a variety of resources, the MPT product database, and other tools to really help bring people together, and it's a resource hub for the field. And if you have other publications or things you want to add, we welcome them. Okay, next slide, please. I also just wanted to ensure that you all are aware of an MPT summit that is being planned right now, entitled Envisioning the Future of Contraception and STI Prevention. The dates are June 24th and through the 25th, 2-day in-person meeting with virtual capacity as well. And this is going to be on the NIH campus. And more information about this is forthcoming. But we just wanted to make sure that everyone here is going to, to know about this and encourage you to come. There will also be a opportunity for poster presentations. So keep that in mind. Okay, next slide. And if you want to have any more information or get back to us on anything, let us know. We're always excited to connect with folks, and encourage you to visit our website and and the database and anything else in our this recording from this webinar. All right, next slide. And yes, we are planning a future webinar. The one coming up is being planned to focus on cervical vaginal microbiome. So with that, I want to thank everyone again, especially the presenters. Your presentations were really stimulating, and I'm sure getting people thinking about the future too. Okay, well, I hope everyone has a wonderful rest of your days. Bye-bye.
Thank you. Bye-bye.
Thank you.